So as a community, we have to find ways to support clinical informatics, genetics assistants basically, to partner in with the clinicians to do these types of analysis and interpret the data. And yes, we'll get better at doing the analysis. Yes, we'll improve the clinical interfaces. Yes, we'll develop through the sort of interface design testing that's necessary to determine that we're making the right calls using the interface. But the bottom line is that it just takes an awful lot of time, and your clinicians are not going to be the ones sitting there at the computer trying to do that. And so we're going to have a cadre of informatically inclined people to work with them.
Yeah, thanks a lot. Yeah, it does seem to say that the future economy is people who do this, and there's absolutely no one, barely anyone working in this field currently, so it's almost totally empty.
If I can just add one other comment, I think it also speaks to the necessity of gels and evaluating how we can best translate and seeing where these problems occur.
I'd be happy to take any further comments from the floor. Beth?
I just thought I'd try to draw a parallel. Sort of when cytogenetics became a regular testing, there's a school of cytogenetics where you get your degree before you're a recognized tester. Then it became molecular genetics, and you would add that. It was, you do 2 years, 3 years, and become a molecular genetics tester. You read the stuff, the results and stuff. Do you imagine that there's eventually going to be a sort of group, cytogenetics, molecular genetics, bioinformatics, for lack of a better word, where there's sort of a trained degree of readers of results? And so they're not developing the tools, but the physician doesn't read them. They deliver the final report, but there's an intermediary between research level and clinic that's a trained, named kind of technician.
I think that's a really interesting point. I could see that. I think it's going to depend. I mean, it might be that we almost have to have that because you could argue that we can make the tests be, ideally, make the tests be interpreted and sort of simple enough that it could be read directly by a physician. But I think one of the reasons why we probably do have to have that is it's going to be a moving target. There's always going to be new things uncovered, and we're probably going to always need that middle ground. And I think that's a really good point with making sure we focus on ensuring that happens, that we don't have this big gap between researchers and physicians, and having appropriate interpretation of results, I think would be become really important.
I'll just add that the software for the so bioinformatics is all about waves of data. And so over a career, I've seen 5 or 6 major waves of data coming from the time that we're doing large-scale EST sequencing through genome projects, through large-scale genotyping, whole genome sequences. These are just sort of a cascade of water that's about to smash down on you when you get overwhelmed every few years with what's coming. And what you see over this process is that it takes about 3 to 5 years for the software infrastructure to mature and become something that's been put in the hands of sort of normal practitioners and not sort of reserved to the sort of research labs that are at the front edge. And so I would expect that in about 5 years' time, this will stabilize. We'll have a fairly reasonable interface for use in the clinic, and there will be something much like Beth Simpson suggested, which is a certification process that goes in place for people who work with this. But how we get from here to there over that 3 to 5 year period is the challenge that we face in the near term.
Okay, thanks a lot. Okay, let's move on to the next question. Let's talk about routine innovation. And where do you see a need for innovative algorithms in bioinformatics? And in what area do you think they need it most? And we'll start with Wyeth this time, I think.
So, for me, the only thing that really matters is transcriptional regulation and gene activity. That's the most important thing in biology. There's enormous need across the spectrum of things that we're doing right now. So right now our attention tends to be focused on genome analysis, and there's plenty of opportunity for new methods in genome analysis and looking at how we look at copy number variation and how we can process that, how we compare large sample sets to one another, how we prioritize candidates of genes. So that work is pretty obvious, and most people recognize that needs to be done. What people aren't talking about so much right now is how do we go in and do large-scale simulations of cells and processes. It's had practitioners working on it, but it becomes the larger challenge as we go along. As we gain the capacity to really study things in depth at affordable prices. And you can say, what are the metabolites that we produce, what are the proteins that are active, what is the system of the cell. Trying to get that into sort of one mathematical model and being able to compute on it becomes a major challenge. And you've seen that happen in other fields, in physics, for instance, where it moves from being sort of a one-person lab to a large-scale lab. And I think you're going to have to see that here. So it's that sort of mathematical biology piece that we're going to have to deal with.
I'll just echo a lot of those comments, or except including just seeing a lot of people struggling with sequence analysis and RNA-seq analysis, for example, is just becoming overwhelming for some people. And we just always need innovation in these methodologies, and but certainly also data integration, visualization of complex and high-dimensional data, I think, are areas where we really need to come up with significant innovation. And then also, sort of systems-based network analysis, and I would say also innovation in organization of bioinformatics software, and how we're avoiding this problem of having lots and lots of methods come out in little pieces in journals and not having a more systematic way of organizing them.
Thanks, Fiona. Any comments from the field? From the floor? Okay, let's take another question in terms of what actually limits us as bioinformaticians. And one thing that the National Human Genome Research Institute in the US recently pointed to was suddenly in the field there's a lack of high-performance computing, and this was actually holding back progress in the field in the way that actually physics and climate modeling are currently basically held back by just the lack of supercomputing to actually conduct their work. Do you agree to an extent that computing is becoming a limiting feature in our field? What kind of things could we do if we had an order of magnitude greater power at our fingers? Or would this just make us more efficient? This was
definitely compute power is an issue. It's always an issue. I think, we could argue it's more of an issue now, but I do remember back when I was much younger, dealing with running a phylogenetic tree and going, oh, this is going to take days and days and days on my one CPU, which was, I think, 66 megahertz or something. And certainly, I think it's always going to be an issue and can always directly translate into making things faster and accelerating research. So I think HPC in support of high-performance computing is critical. I would say in terms of areas that could most benefit, I would say all areas can benefit, but perhaps sort of the protein structure prediction area is certainly one where I see, if we just had an order of magnitude more computing power, we could really get a lot more done, and certainly encourage everybody who has gaming consoles to be running folded on the back end of that.
So I have kind of a mixed response to that. So yes, computing power is always at some level a limitation, but I remember being a limitation when I was 10 years old working on my dad's Concord PET, trying to program that with 8K of memory or something. So I think it's a piece, but it's a small piece. So oftentimes the challenges are, one, having a good idea that will let you figure out how to do something, and that isn't computer limited, it's really people limited. And from a bioinformatics perspective, we have a challenge in that because we are sought after as collaborators and partners by many people. We end up probably working on too many projects per person, and we end up with our focus being a little bit divided. So we're working on interpretation of biochemical diseases, and on promoter design, and on algorithms for regulatory sequence analysis, and so on and so forth. Whereas a more laboratory-oriented researcher will tend to take on one of those projects for an extended period and really focus on it. So we are more diluted than we probably should be, partly because there's just not enough of us as a whole. And then the other reason I'm not sure that computational power is the major limitation for much of what we do. I agree for mathematical biology and protein structures it is. But things like the sequence processing, yes, we're in a period of massive data, yes, we're in a period of challenge, but we're also going to be, given that's going to be sort of a fundamental piece, we're going to see hardware acceleration that's going to emerge pretty quickly that's going to handle an awful lot of the problems very specialized chip that just focuses on genome realignment. And that's going to basically take that whole process out of our dirty hands and simplify it. And so I'm not sure that's the final implementation.
Okay, thanks, Ryan. Happy to take any comments from the floor. If not, we can go on to our next question. So I guess the very young Dustin Hoffman in the film The Graduate was advised to get into plastics. But what do you think is going to be the biggest growth area in bioinformatics in the next 5 years, and what kind of projects should a budding PhD student kind of want to get into? And I guess it's Wyeth.
I'll be fast because I think I've already given a hint to this, which is I think the capacity to work with in a clinical setting with large-scale, high-throughput genome data and interpret the data and deliver that information to the clinical specialists is a huge need. There's going to be opportunities across the world, around the world, and particularly across Canada as we go through this. So if I were entering into graduate studies right now and I was thinking, what should I specialize in? I'd go right there because I know at the end of the training I'd have a nice, high-paying job in a healthcare setting.
Okay, that's why. The right answer is the clinic.
Yeah, I'll actually, I'll add to that. I definitely agree. But the way I actually first saw this question is I wanted to make sure I would want to make sure it's clear to students that whatever they do, think of doing, that they should include incorporating what they are interested in. Don't just go with the next big thing, because you've got to focus on what you're interested in. If you're interested in something, you will sort of naturally excel because you actually like doing it. And certainly it's hard to predict, what's going to happen, say, a decade from now, if you look at a decade earlier. But I have to say, I do think that's an issue. I think there's also the issue of translating a lot of these methodologies and discoveries outside of the clinic in aquaculture and agriculture and environmental research. A lot of these people I'm really noticing are really struggling with wanting to use genomics technologies and are a little bit behind in using them, and certainly there could be a lot of potential there. But it's really important that people focus on what their interests and aptitudes are, and together we can sort of